We are really fortunate to have Dr. Trisha Wittkopp. So it's my pleasure to introduce her. She received Bachelor of Science from the University of Michigan, a PhD from the University of Wisconsin, and she did her postdoc at Cornell University. She started a faculty position at U of M back in 2005, where she's now the Sally L. Allen Collegiate Professor and Arthur F. Thurnall Professor in the Department of EEB and the Department of MCDB, and she's also affiliated in the CCMB. Her research investigates the genetic basis of phenotypic evolution with emphasis on the evolution of gene expression. I checked her CV. All her career, she received many awards. So including the Diamond Running Cancer Research Fellow, Sloan Fellowship, Guggenheim Fellowship, much of them Starter Scholar Award. So instead of spending the time introducing every award of hers, let's just welcome Trisha to deliver the talk titled Molecular and Evolutionary Processes Generating Variation in Gene Expression. Thank you.
Thank you. Thank you for that generous introduction and the opportunity to talk to you today in this new way. So as you said, I am interested in the evolution of gene expression and its role in the evolution of phenotypes. And today my talk is going to focus on gene expression as a phenotype. Here we are. Gene expression is at the heart of biology, in my opinion. It is what it lies right at the heart between converting genotypes to phenotypes. It's what allows cells with the same DNA to create differences in structure and function. It also plays a key role in helping organisms cope with environmental changes and can be both a cause and consequence of disease states. Considering all of these roles, it's not surprising that variation in gene expression also impacts fitness, right, which has longer evolutionary scale consequences. Variation in gene expression is very common. These images here come from a recent study from the GTEx Consortium looking at variation in gene expression in different human tissues and looking at the genetic basis of that variation. So one thing that's emerging from this type of work is that the same genetic variants that are associated with variation in gene expression are also associated with variation in various disease states. So this underscores this idea that variation in gene expression matters to biology, to health, to fitness. And I would argue that understanding the genetic basis of that variation in expression, therefore, is a key step in understanding the variation in the higher order phenotypes. As I mentioned, this also applies over longer evolutionary timescales. So this plot that I'll build up in a couple layers here just shows how as evolutionary time passes, so divergence time increases, which is on this x-axis here, the amount of expression divergence on a genomic scale, and this is just a sort of composite measure, is also increasing with time. And we saw this, we see this clearly in data from different fruit fly species, Drosophila. We see the same type of pattern in yeast. And if you analyze data from human individual tissues, again, we see the same pattern. So there seems to be some commonalities of how gene expression is evolving across diverse species. The goal of our work in this is to really understand the molecular and the evolutionary processes that underlie that variation in gene expression. So it's often described, again, in many studies, but we're really trying to understand why we see the variation that we see. Sort of how is it structured within the genome, within these types of regulatory networks? So where are the mutations in the genome? Do they disrupt cis-regulatory sequences, transacting factors? How do they interact with each other? And then again, on the evolutionary timescale, we want to know about how mutations that generate variation sort of are filtered or sifted through time through the action of selection and genetic drift to produce the patterns of polymorphism we sample within a species and divergence we see between species. And one thing that we like to focus on in my lab particularly is really the interaction between these two ways of viewing variation in gene expression. So what is it about the structure of a regulatory network that makes some types of regulatory variants more or less likely to arise or to survive over time, right, to persist, to be passed from one generation to the next. So in order to distinguish the sort of evolutionary causes for variation that we see, we often use a neutral model. So if we're talking about sequence variation, which is, I think, a more common framework for many people, we can look at the variation that exists within a species or between species, and we can use our understanding of the evolutionary process to predict how much variation we would expect to see under neutral evolution, so in the absence of natural selection. And we can compare that to what we do see to infer whether or not selection has shaped the variation in that sequence. For traits, for quantitative traits generally, and gene expression is a quantitative trait, this type of framework also applies, but has been more challenging because that neutral model, the knowledge of what happens in the absence of selection, how much variation do we expect and what types of variation is less clear. If, however, we can get that, if we can define a distribution of mutational effects, and so these are just two hypothetical distributions drawn here, so if we can know how new mutations are creating, say, variation in gene expression, and we can compare that to the effects of variants we see in natural populations, polymorphisms, if the distributions are the same between mutations and polymorphisms, we have no need to invoke selection to explain that variation. We can say that that variation seems to be consistent with just a random sample of new mutations that are arising sort of spontaneously. If, however, we do see differences between these distributions, we can infer that selection has acted and even say something about how it's acted. So in this cartoon example here, we would say that selection has favored individuals with higher trait values or disfavored those with lower trait values, causing the polymorphism we see, the variation in a natural population, to deviate from what would happen if only mutation and chance were responsible for that variation. Okay, so this is a simple framework. Why aren't we doing this? And as I said, it's hard to get that mutational distribution. So let me just back up a moment and say that was what one of the things that I set out to do in my lab when I started here back in 2005. So it's been a longer term project that I'm going to give you some highlights from today, pulling this together. So we started with what I consider some pretty straightforward and basic questions. How often do new mutations affect expression of a particular gene, right? Of all the mutations that arise, what fraction would affect expression of gene X? How big are their effects on expression and in what direction? Do they tend to most often increase expression, decrease expression? Do they change it a little? Do they change it a lot? Do different types of regulatory mutations, cis and trans regulatory mutations, for example, arise with different frequencies? Do they have different distributions of effects? And what impact do these mutations have on fitness? Because of course, that's the ultimate determinant of whether or not they're passed to the next generation and survive to be sampled in the sort of real world populations. I would argue that at least when we started this work, all of these were very wide open questions, not really known for any gene in any species. So why is this challenging? Why is it hard to get these neutral expectations from the effects of mutation? Well, number one, mutations are rare events, right? So they happen all the time in all living things. But if we're asking about mutations that affect expression, as we are in our case of one particular gene, we expect those to arise pretty rarely. So if we're going to characterize them, we have to be able to sift through, millions or hundreds of millions of events to find them. Sensitivity, we reasoned that most new mutations, and this is consistent with, I think, observation, should have pretty small effects. And so we not only need to be able to search through a lot of mutations, we need to be able to do it in a very sensitive way to pick up potentially small changes in expression. And then finally, again, we need to do it at scale. We can't describe a distribution with only one or two mutations. We need to be able to get hundreds, if not thousands, to start to understand the shape of those distributions. So again, back in 2005, when I was thinking about these questions and wanting to find a way to pursue them, we figured out ways to kind of overcome some of these hurdles. So to deal with the rarity of events, we decided to elevate the mutation rate in a controlled way using a chemical mutagen EMS. And this has the benefit that we can control the exposure, dose, and time to elevate the rate enough to make collection feasible, but not so much that we sort of start getting into problems with multiple mutations. For sensitive detection, sort of quantitative small changes in expression level, we turn to reporter genes, fluorescent reporter genes that we can analyze with flow cytometry in individual living cells, where we have the power to detect changes in expression as small as 1% consistently. And then for scale, of course, a high throughput scoring mechanism, and that's again where the flow cytometry comes in, makes this feasible. So the experimental system in more detail is here. Gizem Khaleh was my very first graduate student, and she really built this first strain and got the system up and running. It's a simple model. It has we start with a haploid yeast cell, Saccharomyces cerevisiae, and we have a reporter gene integrated into the genome that contains the promoter from the TDH3 gene. TDH is a gene from Saccharomyces cerevisiae, from the baker's yeast, that encodes a glyceraldehyde 3-phosphate dehydrogenase. So it's just the gene itself encodes an enzyme involved in glycolysis and gluconeogenesis. And in this part of the experiment, we're just using the promoter from it. It's a well-characterized promoter. It drives high expression that's constitutive throughout the cell cycle, simplifying the experiments. And then our fluorescent readout here is a yellow fluorescent protein. So we take a population of these haploid cells, and we used haploid because we're, again, trying to recover mutations that affect gene expression, and we didn't want to miss recessive mutations. So we started with the haploid cell. Split the population of these cells into two, treated half with that chemical mutagen EMS. And I'll point out that EMS does not capture the full spectrum of spontaneous mutations. It introduces only point mutations and almost exclusively G to A and C to T changes. Those are, however, the most common spontaneous mutations in yeast and the most prevalent polymorphisms that you'll find segregating the yeast population. So if we had to pick a subset, we felt it was a pretty good subset to pick. So we had these two populations. We first, analyzed them with flow cytometry, and what you see here is the distribution of the two populations. There are actually two curves. They're hard to see. One is red dotted and one is a black line. They're hard in part because they almost completely overlay each other, and that's by design. The dose of EMS used was enough to increase the number of cells in the 1% tails of the control population to contain 2% of the mutagenized cells. So again, we're trying to balance this tightrope of increasing mutations enough to make it feasible to find mutants that have altered expression, but also keep it low enough so that when we find such a cell, it only has one mutation that is affecting this gene's expression. From follow-up work, we know that this dose of EMS that we settled on introduces about 30 mutations, 25 to 30 mutations per cell in all. But again, only one is affecting expression of our focal gene. So in this first experiment, we sorted from the tails of this distribution, putting individual cells into spots in a 384-well plate. We can then grow them up clonally, inoculate new liquid cultures, reanalyze those, and what we're looking for are cases where we see a statistically significant change in fluorescence in that genotype. So again, in this first round, we isolated 231 mutant genotypes that had changes in fluorescence and subjected them to further characterization. Specifically, we wanted to know what type of mutation in each of these was affecting fluorescence or expression of this gene, and what was that effect. So I want to pause for a minute and remind you the broad classes of mutations that would affect a gene's expression. So here's our cartoony gene. Its coding sequence would be transcribed into RNA, protein. In this case, that would be the YFP. We have a promoter, so our cis regulatory sequence, and changes in that can affect the gene's expression. The promoter is interacting with transcription factors whose activity is influenced by a whole host of indirect factors that influence its activity. So we can consider mutations that affect this gene's expression as cis-acting or trans-acting, and that's a broad inclusive class here. Okay. We can also get changes in expression or changes in fluorescence that we detect if the reporter gene is duplicated or deleted. And so we also needed to consider that. So we broke them into four classes, initially by resequencing the reporter gene in each of those 231 mutants. So if we found a non-synonymous change in the coding region of YFP, that was considered a coding mutant. If there was a change in the promoter, is 678 base pair promoter. That was a cis regulatory mutant. If there was a change in the copy number of the reporter gene, that was a CNV. And if there was a heritable change in expression, but no changes at all in our reporter gene, that was a trans regulatory mutant. It's somewhere else in the genome, was the class. So we wanted to know first about the relative frequency of these. And what we found was that most of them were transacting. That's not necessarily surprising given the size of the rest of the genome, the genome outside of the reporter gene. It's a much larger mutational target size. However, remember, these are selected not just for mutations happening in different regions, but mutations that affect expression of this gene. So although we could have predicted trans would be more common, the relative frequency was, I think, we couldn't predict. We recovered some mutations in the coding sequence. There were 22 copy number variants, but we found them at similar rates in our control population. So these are not induced by EMS. This is just what happens when you grow yeast cells in the lab. They have spontaneous aneuploidies and even whole genome duplications. And that 22 is 11 of each of those. And then 4 mutations in the promoter. And this is regulatory region. So as I said, we also wanted to know how these different classes of mutation might differ in their effects on gene expression. So we looked at the 4 cis regulatory mutations, and I should point out how to read this plot. So this is a Z-score. So positive values are increases in expression, negative are decreases in expression, and then we have the frequency of mutants with those types of effects. So the 4 cis regulatory mutations, 2 increased expression and 2 decreased expression. The coding mutations, all of them decreased expression, and about half of them decreased it a lot. Now, YFP is a highly optimized, tool in the lab, so it's not surprising that virtually any mutation in it would decrease its fluorescence. The copy number variants all turned out to be either duplications of the chromosome carrying the reporter gene or whole genome duplications. And so consistent with that, most, granted not all, there was one exception, of these increased expression. Now, the transacting class, going into this, we were much less sure what to predict. I'm not sure I said yet, but TDH3 is one of the highest expressed genes in the yeast genome. If you rank the 6,000 genes in yeast from highest to lowest expressed, it comes in as number 4 or number 6. So it's really highly expressed. And that alone made me guess that most of these new mutations would be decreasing that gene's expression. In fact, that's not what we saw. In this first experiment, there was actually a slight majority increasing. We've repeated this experiment many times over, and I can say it's either symmetrical or there's a lean towards increasing, but it's definitely not biased towards mutations that decrease expression. Okay, now I mentioned that one of the things we really wanted to do was know how the properties of these mutations differed for cis and trans regulatory mutations. Now with 4 cis regulatory mutations, that's not much of a distribution to compare. So we backed up and a former graduate student, Dave Yuan, created a set of cis regulatory mutations. This is again, before some of the ways we could do this today, with kind of a brute force site-directed mutagenesis, marching along the promoter, changing every G to an A and C to a T to mimic the effects of EMS. We could then look at the effects of each of these changes. Those are shown down here below. And the ones that had the largest effects are, you can see that the big red lines, those fell in 3 previously characterized transcription factor binding sites. So the binding sites that were already known do seem to be controlling most of the activity of this promoter. However, there are other mutations that certainly have effects, and here's a cluster that may be a binding site for another factor that just hasn't yet been identified. So we can then, so this is how we increased the size of our cis mutant collection. For the trans regulatory mutants, excuse my dog, this is the fun of working from home. We repeated the experiment, except instead of sorting from the tails, we just decided to sort randomly. And we did this in part because we had introduced the cis mutation sort of systematically without any selection for extremes. And we wanted to compare it to a trans set that was isolated in the same way. So this time we isolated almost 1,500 independent genotypes and assayed their effects on fluorescence, on expression. And those 2 distributions for the cis mutations and trans mutations are shown here. So again, this is trying to understand how both of these classes of mutations affect expression of the same gene, but are there systematic differences in those effects? That's what we wanted to know. So at the top is the distribution of effects of those 236 cis regulatory mutations that I showed you that we marched across the promoter. The ones highlighted in black are in those three well-known binding sites, transcription factor binding sites, and they have big effects, as I showed you before. Everything I'm about to say after this is true whether we include or exclude those black data points here. What you might already be able to see if you're looking at these two distributions is that the effect size of cis regulatory mutations tends to be larger than trans regulatory mutations. And I think we can see this even better if we essentially fold these distributions in half and focus on the magnitude of change rather than its direction, rather than increase or decrease in expression. So again, we see that cis regulatory mutations tend to have larger effects on the gene's expression than trans regulatory mutations. We were also interested again in comparing the relative frequency of these classes. But to do this, we had to take into account that, again, the cis mutations were targeted and the trans mutations were random. So taking that into account, we could then compare kind of the relative frequency of cis and trans regulatory mutations. And we discovered that it was important to do that, taking into account their effect size as well. So let me just say, if we consider mutations with any effect on the gene's expression, including no, so basically the potential target size for cis or trans regulatory mutations, this is much larger for trans, so trans is in blue here, than cis. However, if we ask about the relative frequency of cis and trans active mutations that, for example, decrease this gene's expression 10% or more, cis regulatory mutations are actually more common. We've done screens where we look to try and enrich for trans regulatory mutations that decrease expression at this range, and we can't recover them, whereas we readily get the cis regulatory mutations. At the same time, if you ask about mutations that increase expression, trans regulatory mutations are much more common. Again, we can't find any cis mutations that are increasing it to this degree. So cis and trans regulatory mutations are arising at different rates. Sorry, I'm sort of going out of order from my slide here, so let me back up so that we're aligned with the text. These data showed, number one, that if we're talking about mutations that affect this gene's activity, meaning change its protein coding sequence or its expression through regulation, we see that regulatory changes arise much more frequently than coding changes. So that may be one reason that they're such a prevalent source of variation, phenotypic variation. Cis regulatory mutations tend to have larger effects than trans regulatory mutations. And on the flip side, trans regulatory mutations are more common overall, but that relative frequency does depend upon the effect size and direction. I also didn't show you the data, but most new trans regulatory mutations we've shown by comparing the effects in diploid cells are recessive, whereas those cis-acting mutations are not. So these differences in frequency, effect size, direction, dominance may all be contributing to cis and trans regulatory mutations having a different evolutionary fate, how long they survive within a natural population to be sampled, for example. Okay. Now, the molecular biologists or people that think more about molecular biology may be wondering, what are all these trans regulatory mutations? And so we've spent a few years mapping that. So as I mentioned, the mutations were introduced with EMS, and this is about 25 to 30 mutations per cell. So sequencing them alone does not tell us which mutation affected the gene's expression. We have to actually do a genetic mapping experiment, recombination, bulk segregation analysis, and sequencing to get to that. But we wanted to know the identity of these mutations so we could answer questions like these that I'll go through on this slide. We had questions about how predictable or how predictive a regulatory network could be for understanding the sources of a gene's expression variation, and how they might be structured within it. So this network that you see here was inferred from published data in yeast. A lot of it is ChIP-seq type data that's connecting a transcription factor to a particular region of the genome. These are all, with the exception of TDH3, on this map transcription factors. And so what you see here is what I think of as two layers in this network. So we have genes that were predicted to directly regulate TDH3, including RAP1 and GCR1, which bind to those binding sites I pointed out previously as well characterized, as well as factors predicted to directly regulate those first level factors. So it's sort of a two-step network here. We also wanted to know where these mutations fell within genes. Did they tend to hit coding sequences or non-coding sequences and therefore affect regulation? Were they clustered within the network? Is there a certain part of the network that was more likely to have mutations arise that affect expression? Or were they dispersed throughout the network? And did they tend to be enriched in direct regulators or not? So these were all kind of questions that our unbiased sampling of mutations allowed us to look at. So Fabien Deveau is a former postdoc who developed the mapping strategy and did the work. And I just want to underscore here that the effects we're mapping, these mutations, are changing expression as little as 7%. So if you're thinking about kind of more RNA-seq data, this is a level of sensitivity that's sort of almost a different magnitude. So he was able to identify 61 point mutations that mapped to 39 different genes spread over 60 strains. So what that means is one of our strains had 2 mutations in it that both were associated with expression. The fact they mapped to 39 genes means that some genes were hit multiple times. So while I don't think we've hit every gene that could affect this gene's expression, we're also not hugely undersaturated, right? So there does seem to be a defined set of genes that are most likely to carry mutations that affect this gene's expression. We also discovered some duplications of other chromosomes that then had transacting effects on expression of the reporter gene. Okay. Not only did we map these mutations, another nice thing about working with yeast is we can go in and engineer a single nucleotide change to test the associated variant and show that, yes, it actually does cause the change in expression that we inferred from the genetic mapping data that it did. So down at the bottom is the effect of the mutation, the effect on expression of the full mutant genotype with the 25 or 30 mutations. And on the y-axis is the effect caused by that one mutation we mapped. So you can see that in all but one case, basically, it largely explained it. Okay, so again, what are these mutations? Where do they map in? Only 3 of our mutations fell within genes that were in that network I showed you, that were in what we consider the predicted TDH3 regulatory network. And those 3 mutations mapped to 2 different genes. So one of the genes was hit twice. There was another cluster of genes, essentially all of those that increased expression 10% or more, hit one of 4 genes that are all involved in adenine biosynthesis, purine biosynthesis. So 21 different mutations fell. I guess I have 5 genes. I've only got 4 listed there. I think I forgot to add one. So there seemed to be some predictability. In fact, we didn't find all of those by genetic mapping. Once we had done some of the mapping and saw this recurring pattern, we went back to our data and said, okay, let's just sequence these genes in every genotype that has an increase in expression more than 10%. And basically we found the mutation, right? It was a non-coding change, a non-synonymous change in one of those genes. There was another set of genes involved in copper or iron transport where we found 7 mutations that mapped to 5 genes. So again, some predictability, some consistency in the source of this variation. The rest of them fell in basically genes associated with the normal geo categories that you get when you do these types of analyses: transcription, post-transcription, chromatin, glycolysis, cell cycle, and others. So again, going back to the questions I posed, where do these mutations fall within the network? Well, 2 of them hit 2 genes, 3 mutations fell in these 2 genes, ROX1 and TI7. We did not recover any mutations in these direct regulators, RAP1 and GCR1. We went back and actually did kind of a full mutagenesis of the coding sequence of each of those genes and just asked, could we even recover them, or why might we not have found them? And for RAP1, we think we didn't recover them because RAP1 mutations are either lethal or viable, but don't have an effect on TDH3 expression, so therefore wouldn't have been pulled out. GCR1, the effects it's a little bit more equivocal. There is some lethality explaining part of it, but it's environment dependent, and we think that might have just been, mutations were missed because of the sample size. All of these mutations, these transregulatory mutations, map to coding sequences in the yeast genome. Now, as somebody who has spent a lot of time studying non-coding sequences, and particularly in fruit flies, which is what the other half of my lab works with, this was surprising and I have to admit a little disappointing, but the yeast group, yeast members of my lab were eager to quick to remind me that the yeast genome is not the fly genome or the human genome. The yeast genome is 75% coding sequence and only 25% non-coding. So there's certainly a lot more opportunity in yeast for these mutations to be in coding sequences. As I mentioned, there was a lot of different genes mutated, so they are sort of dispersed. However, there were 2 pathways that were hit multiple times. So there is some predictability or clustering, if you will, as well. We didn't recover mutations in the known direct regulators, as I said. So we got these indirect regulators and things that aren't transcription factors in many cases. Okay. I said at the outset that one of the main motivations was to understand the effects of mutations so that we could compare them to the variants that are in natural populations. So we were able to do this here because a separate study within the lab, so Brian Metzger, a former grad student, had done a highly powered EQTL mapping study between actually 3 different pairs of yeast strains. So these are the 3 different pairs here in red, yellow, and blue. And each of the regions that you see this colored bar is a QTL that we mapped affecting expression of TDH3. And so this had multiple rounds of recombination, and so has much higher resolution than other EQTL mapping studies in yeast that usually find about 8 to 10 EQTL per gene. We found about 100 in each of these cases. And it's closely linked EQTL that the recombination starts to break them up and you see opposing effects. And so when we looked at how the causative mutations that again came out of this EMS screen compared to those regions of EQTL, so essentially, does our understanding of the mutational piece give us any predictive power for where naturally occurring variants may lie? We found that there was indeed enrichment in those regions, suggesting there is some predictive power. Okay. So as I said, we really wanted to characterize mutations so that we could use it to understand that variation in natural populations. So the next bit I'm going to tell you about is using that, that comparison to focus on the cis regulatory sequences, so mutations in the promoter of TDH3, and comparing the effects of mutations to polymorphisms. So again, if we see no difference between those 2, we have no evidence to say selection has shaped that variation. If we do see differences, we can say selection has had an effect and something about that effect. Okay. The polymorphisms that we sampled came from a set of 60 different Saccharomyces cerevisiae strains that have been isolated from a variety of environments. So you can see kind of bakeries, distilleries, wineries, nature like tree bark, and so on. And so we found that these strains, number 1, contain variation in TDH3 expression. A large portion of that was due to cis regulatory variation, and we were able to show that it's due to genetic variation in the promoter. So that's the portion of this variation in this population that I'm going to focus on here. And this next little bit will really highlight work led by Brian Metzger, a former graduate student in the group. So the first thing that we did was to look at the promoter itself and just sequence it and ask, okay, among these strains, how many sequence variants are in the promoter? And how does that compare to the number of sequence variants that would be silent changes in the coding sequence of TDH3? And those silent sequences, those silent changes rather, are considered a baseline for neutral evolution. So again, if we see, as we did, fewer variants in the promoter, then there are segregating silent changes in the coding sequence, then that's a sign that selection has acted on this promoter to eliminate some of the genetic variants. There's been negative purifying selection here. It doesn't tell you why variants were rejected. It just says that there's less variation here than we would expect by chance alone, okay, by mutation and chance alone. So as I mentioned, we had measured the effects of mutations in this promoter. We used the same system to isolate polymorphisms and test them in parallel. And what we found is that the distributions of effects of those 2 were not significantly different. So from these data, we had no evidence to say that selection had shaped this variation in the promoter for its effects on average gene expression, so the sort of mean level of expression among genetically identical cells in this case. Now that was a little surprising. TDH3 is a highly expressed gene. It's possible that maybe variation in its expression doesn't affect fitness, but we wanted to test that. And so we were able to do that by taking the variants that we had originally put in the context of the promoter and a reporter gene to measure its impact on expression and moving them to the native TDH3 locus such that they're now affecting expression of TDH3 itself and potentially having fitness consequences. We then compare the growth rate of that cell to a reference genotype, and just by the relative frequency of the two populations over time, we get a direct measure of their relative fitness. Okay, so we did that for mutations that caused a range of changes in expression of TDH3. So the effects of the mutations are shown here on the x-axis, the change in fitness is shown on the y-axis. And I want to pause here because I think I now take this for granted, but we don't always know how much, a 10% change in a gene's expression matters or at all. And so I think this is one of the rare cases where we can really see that in great detail. So a complete loss of function of TDH3, that's our 0% mutant down here, causes a 7% reduction in fitness. If you're not an evolutionary biologist, 7% might not sound like much. However, over evolutionary timescales, that's a really big effect. And if you happen to have a mortgage and you think about an interest rate of 7%, you can start to see, or you maybe appreciate better that compounding that can happen. So you can see as you move towards the wild type expression level, which does in fact have the highest fitness that we observed, you have an initially kind of linear and then sort of plateauing kind of increase. We see reduced fitness for both decreases and increases in this gene's expression. So there is an optimum. Okay, so we can now use our understanding of how changes in this gene's expression affect fitness to look again at the mutations and the polymorphisms. And what we find is that most mutations that occur in this gene's promoter don't cause changes in expression that are great enough to cause much of a change in fitness. And so if mutations are arising, most of them are not going to alter the average expression level of this gene enough for selection to act on them. Not that the change in expression of this gene doesn't matter, it's just that mutations that have effects that would matter are very rare. So it's a frequency-based observation. Okay, but I told you that we can see signature of selection on this promoter because we see fewer genetic variants. So how do we reconcile that? Well, Brian decided to take advantage of the richness of our data. So let me back up and say a little bit more detail what we're doing here. So when we measure the effect of a mutation on expression of our reporter gene, what that actually looks like is this, where we start with up to 12 replicate populations, each having about 10,000 cells in it. We subject them to flow cytometry. The flow cytometer gives us a measurement for each individual cell that estimates actually the size of the cell as well as the fluorescence, and then we correct for cell size differences. So we don't get a single measure of expression, but rather we get a distribution of expression phenotypes from which we calculate average expression. And in fact, we can calculate the variance of that distribution, which is related to a quantity commonly known as expression noise. So this is the differences in expression among genetically identical cells that occur even in the same environment, okay? Expression noise is actually heritable, so different genetic changes can change noise. And what we found when we compared the effects of mutations and polymorphisms was now there is a difference, and specifically, there are many mutations that can arise in that promoter that we don't see in natural populations among the polymorphisms, and that this deviates in a way that suggests that mutations increasing expression noise are selected against. They tend to be removed from the population preferentially. So this again suggests that changing expression noise, or this variability aspect, impacts fitness. So we wanted to know, to try and directly measure that. Now, this has been suggested by many people that noise may matter and may affect fitness, but it has been really hard to tease that apart and directly measure, in part because nearly all mutations that alter expression noise also alter mean expression. And I've shown you that both potentially may matter. So these plots here, or these points on this plot, are the initial set of 236 cis-regulatory mutations that we made. And you can see those that cause dramatic changes in mean expression also cause correlated changes in expression noise. And so we needed a way to decouple the effects of those two. So Fabian created another couple hundred variants or alleles of this promoter. And here he was introducing binding sites, combining sets of mutations, just kind of doing everything he could to disrupt this promoter, mutating the Tata box. And what this did was create a set of alleles that varied in both mean and noise. And we were then able to use them to tease apart the relative effects of those two properties on fitness. We've done this in a more complex way in the paper. I'm just going to give you the simpler way here, which shows the same result, which is to say, let's consider pairs of genotypes, such as this red and green shown here, that have the same mean expression, but differences in noise. And then we're going to grow them head to head in that competitive growth experiment to measure their relative fitness. So when we can consider the first two, so here we have genotypes that are both reasonably close to 100% or wild type expression levels. They have some differences in expression noise. What we saw in those cases was that the noisier genotype had lower fitness. In other words, higher noise was bad. Being noisy was bad in those cases. When we looked at 3 other sets of genotypes, however, we found that the noisier genotype didn't always have lower fitness. In fact, the further we moved away from the wild type expression level, we started to see that being noisier was actually beneficial. The noisier genotype had higher fitness. And we've since done simulation experiments to really kind of say that this actually does fall out from first principles, and at the same time was not necessarily what we expected. So I'm just going to give you the kind of intuitive interpretation here, which I think is, at least for us, even though we didn't expect it, once it clicked, felt rather obvious. So if we have 2 genotypes shown by the red and the blue, these would be the distribution of expression among genetically identical cells in the same environment. The red genotype has higher variance, higher noise than the blue genotype, but they have the same mean. If those 2 genotypes have a mean expression that's close to the optimum, the noisier genotype, the red one, has lower fitness because you have more individual cells that are further from the optimal expression level. When, however, your mean expression is farther from the optimum, the noisier genotype is actually better, is beneficial in evolutionary terms. And it's because the tail of the red distribution that's closer to the optimum, those cells divide at a faster rate that actually outpaces the cost of the cells at the other tail of the red distribution. Okay. So higher noise can be either beneficial or deleterious depending on where you are on this kind of fitness landscape. Now, one could ask why you would ever be over here far from the optimum, right? If selection is happening in natural populations, that should be working to keep phenotypes near the optimum. One possibility is you can, of course, have an extreme bottleneck that fixes a deleterious variant. Perhaps a more common one is a change in environment that changes what the optimum optimal expression level is, right? So this change in environment. So to explore that possibility, we changed the yeast environment, and we did that by growing them on different carbon sources, media containing different carbon sources. So everything I've told you before had to do with data that came from yeast growing in a glucose-based media where they undergo fermentation. In this experiment, we also compared the effects on expression in galactose, glycerol, and ethanol-based media. Okay. What we found was that when cells are growing in these different media types using these different carbon sources, the same level of TDH3 expression does indeed have sort of different fitness consequences. So I've showed you before what you see here in red, which is that glucose fitness curve. I've shown you that a couple times. The other lines are the equivalent experimental data done in different environments. So for instance, 100% of wild type expression of TDH3 and glucose is the optimal level for growth. However, that's not true if the cells are growing on galactose. That would be a lower level would be needed for the optimal growth, or glycerol or ethanol. Now, this suggests that if you were shifted to galactose, having noisier expression might be beneficial because by chance you'd have some cells that would have expression closer to this optimum. So that may happen in some cases. However, if an organism is frequently experiencing different environments, and yeast do, there's actually a better solution, sort of in evolutionary terms, and that is plasticity, right? And we know that plasticity is common, that when you change environment, gene expression changes. And so we looked at that plasticity of the wild type TDH3 allele. So in glucose, we just define that as 100% or wild type expression. And we see that actually that wild type allele, when we shift it to growth on galactose, its expression decreases. And if you look at the fitness curve, you can see that that decrease in expression is advantageous. It makes the cells better fit for their environment. That's just that degree of plasticity there. We did the same trick of comparing the effects of mutations and polymorphism, now looking at the effects on plasticity, so that difference in expression between glucose and galactose. And again, found evidence that selection was acting on these genetic variants, disfavoring those that changed plasticity. So there are lots of mutations that were arising that caused the plasticity between these environments to differ, but those were not represented, many of those were not represented in the polymorphisms, the variants that we actually see in the wild. Again, consistent with them being beneficial, a certain degree of plasticity being beneficial. When we look at the plasticity that we see in glycerol and ethanol, we again see different decreases in expression of the wild type allele, but to a lesser extent. And you see that that's actually not adaptive, suggesting that our fitness curves don't suggest that would be adaptive, and that there's a variety of reasons that might be. So putting this all together, I hope today that I've convinced you that understanding the evolution of gene expression is important and is a key component of understanding the evolution of phenotypes. That a neutral model that's informed by empirical data, understanding how new mutations affect a trait, is really powerful for disentangling sort of neutral and selective and neutral and adaptive kind of effects. That gene activity seems to be more often affected by regulatory than coding mutations, just from kind of a new mutations arising perspective. So there's more opportunity for variation in regulation than coding sequences, I would argue, based on these data. Transregulatory mutations arise more frequently than cisregulatory mutations, but tend to have smaller effects, tend to be recessive. Again, these may all be factors that help explain why we see the patterns of cis and transregulatory variation, say contributing to, acting as in EQTL data sets, contributing to expression variation that we see. And then finally, selection acts on regulatory mutations, and we've been able to really carefully define multiple reasons or multiple ways in which selection can act. So mutations that change the average expression level of a gene can impact fitness. Mutations that change the consistency of that gene's expression among genetically identical cells in the same environment can also impact fitness. Plasticity, so the degree to which a different mutation allows expression to change between environments, that's also something that selection is acting on. And then I didn't get into this for the sake of time, but the most recent batch of data from this project in the lab is a grad student, Petra van de Zand, has been studying, is really looking again at those transregulatory mutations to understand the consequences of pleiotropy. And so just very, very briefly, I think I have a minute to briefly say this. This bottom plot here, that gray curve, which looks flattened out because of a change in the y-axis scale, is the same as the gray curve up here. And the points falling away from that curve, those are transregulatory mutations that affect expression of TDH3, but also presumably other genes in the genome. And so the deviation from that gray line is giving us a peek at the consequences of pleiotropy on fitness, which is something that's often discussed, but has been very challenging to actually tease apart and quantify and measure. So we're excited to be looking at that. And we've also done a lot of RNA-seq analysis now on these, so we can say a little bit more about the molecular structure of what's happening in terms of the pleiotropic effects of these transregulatory mutations. So this was collaborative work. I tried to highlight a number of key players along the way. I will underscore Brian Metzger, a former grad student, Fabian DeVoe, a former postdoc, Dave Yuan, a grad student, Andrea Hodgins-Davis, former postdoc, Bing Yang. I didn't have a chance to go into too much of his work today, and Jonathan Gruber. This was a group that really brought a lot of that to life. And as I said, now Petra van de Zand is really digging into the pleiotropy piece. So with that, I will thank you for your time and I'd be happy to take any questions you might have. Stop sharing.
Oh, thank you so much, Trisha. So do we have questions from the audience?
I have a question, Gil.
Yep.
Very nice work, very nice indeed. Now, this is all looking at sites of mutation and transcript expression. Have you ever examined the further dimension of the change in expression or functional consequence at the protein level? Is that of interest?
It is of interest. It wasn't our focus of this work, right? We were really intentionally, swapping in a reporter gene so that we could interrogate the regulatory network. That was the focus. I have a current postdoc in the group right now, Mo Sadiq, who is very interested in kind of the interplay between regulatory and coding changes. And so he's now starting to look at how changes in the protein coding sequence for TDH3, which can impact its enzymatic activity, how those compare to the properties of these regulatory mutations that change its abundance. So it's something we're exploring now, but I don't have data to speak of at the moment.
Yeah. Another question while I have you. Splicing. So in mammalian systems and flies too, I'm sure, alternative splice isoforms are an important source of protein and genomic variation. In yeast, it's quite uncommon. I take it the strains you use don't have any significant splicing.
The gene that we've been focusing on as our readout is not spliced. There's no introns in it. So I can't say anything about splicing at other genes from these data, although I guess our recent RNA-seq data would have some of that in it, but it hasn't been a focus of this work. Yeah.
Great, thanks.
Can I ask a question? Can you hear me?
Yes, Margaret, thank you.
Yeah. So I'm interested in humans in plasticity genes, and what we call plasticity is a little bit different than what you call plasticity or what behavioralists call plasticity, in that you called it noise. But how could it be that it's the same as what we call in human plasticity, which is basically environmental response? So plasticity means you, in behavior, for example, you're more responding to the environment versus less responding to the environment. So the plasticity alleles are actually not noise. They are more responsive to the environment. And you try to keep your environment the same, but there are environmental factors I'm sure that you are not aware of, in which case it would explain it's a different explanation than what you gave for why a plasticity allele might have actually superior property.
So I actually think we don't disagree, and I apologize if I wasn't clear enough about making the distinction. So, we did look at plasticity by intentionally shifting environments, right? And I think that that's consistent with what you're talking about, right? So we'll put that that's a separate set of experiments. But I think what you're arguing is that when I said expression noise, and I described it as genetically identical cells in the same environment, I think you're pointing out there may be microenvironment differences. We put the same, we squirt media in a flask and they grow, but of course, some cells near the top or the bottom may have, we aerate, but anyways, there may be differences. What I would say and why we are not capturing that and why we are actually looking at noise is that I guess I should be a little more cautious here. We can take different genotypes and grow them in the same media, which presumably would have the same range of microenvironment variation. And we see consistent heritable differences in the variance of expression within that. So there's a heritable component that's controlling the variance of that gene's expression. So if it's interacting with microenvironment variation, or it's just the stochasticity of gene expression, which is what we think it is, right? That you have transcriptional bursting and differences in burst size and burst frequency that generate just, again, stochastic differences among cells. That's what we think we're capturing again, and we see the heritable differences for. Thanks.
I have, hi, this is Damien Furman. I have one question related to TDH3. You said that TDH3 is one of the most highly expressed genes in yeast, right?
Yep.
So, to me, that suggests that this is an essential gene for survival of the yeast. So if you're going to have a mutation, if you're going to tolerate a mutation, you probably go for one that would enhance expression of it rather than reduce expression or silence expression, right?
Yeah.
So I'm just wondering, like, if you used your approach on a gene like that had like middle of the road expression or less essential, like, do you have you done that, and what have you seen happen in that situation?
Yeah, so I can partially answer your question. I had a hidden slide here. What we have done, so let me just say that the reason we started with TDH3 was, number one, the promoter had been well characterized. There were some previously characterized binding sites, so we had some hope of interpreting the variation. The other thing was that we did need a certain level of expression for reliable detection and quantification with the flow cytometer that we were using. So we kind of needed to be on the upper end of the expression scale for that reason. And then, of course, get the sensitivity for mutations that increased and decreased expression. We have since gone back and repeated sort of the effects of mutations, that EMS experiment, with reporter genes driven by, I think we have 10 promoters here, although TDH3 is included as well as a comparison. And what you're seeing on the left-hand side are those distributions of mutational effects for these different genes, so with the same assay. These genes don't span the full range of expression levels for the reason I mentioned. We're really limited technically to looking at the top 10 to 15% of expressed genes, again, for detectability. But one thing I think that's interesting here is you can start to see the gene-specific properties of these distributions, which I think are reflecting differences in the regulatory networks controlling expression of these genes. On the right side is if you just sample randomly from these distributions and project variation changes in expression over time, what those differences in the distributions on the left would do to expression. So for instance, you can see that some are more narrow, mutations tend not to affect expression, some are wider, some are sort of skewed one way or the other. It doesn't fully answer your question, I think, but I hope it speaks to it.
Yeah, it does speak to it. Thanks.
Great. Okay, well, my dog is announcing that we're done.
Okay.
Sorry, no, it's 5 o'clock. So thank you all again for your time, and this was fun.
Oh, thank you so much.
Thanks. Bye.
Um,
Thank you.